Thank you. Thanks, everybody, for showing up on Sunday morning. And I'm very happy for this opportunity to share with you some of the work I did together with other people at the lab of Yaniv Ziv at the Weizmann Institute. I thought I'd start with a story about a person that sees a huge elephant that is tied with a rope to a small stake, and it's very unclear what is the rationale for that, because it's very clear that this huge elephant can escape if it wants to. And then he asked the owner of the elephant for the reason for tying him up for this small stake. And then the owner explains that when the elephant was young and much smaller, the same stake and rope were used, and at that age it was enough to hold it. And as the elephant grew up, it was conditioned to believe that breaking away is impossible. So why am I telling you this very American motivation story? System neuroscience as a field was born once people start being able to look into the brain, see the patterns of neuronal activity, and try to decipher their meaning and their relevance to things like behavior or cognition. And over the years, the field has slowly matured, and thanks to methodological and technological advances, larger and larger numbers of neurons could be recorded simultaneously. And my fear is that despite the growing number of simultaneously recorded cells, it will be hard for us as a field to escape from the conceptual framework that was developed when the field was much younger and constrained to a limited number of simultaneously recorded cells. So the key tool for interpretation neuronal data that enable multiple seminal findings over the years has been the so-called neuronal correlate approach, and specifically the calculation of a neuronal tuning curve that captures the specificity of the neuronal response to the examined external variable, like this famous example from the seminal work of Hubel and Wieser. However, obtaining this kind of tuning curve requires some knowledge, or at least assumption, about the identity of the encoded variable, which in this case is, of course, the orientation of the visual stimulus. And then we saw that the neurons in different brain regions encode different variables. However, sometimes we simply don't know what variables are encoded in the brain region of interest, and even if we think we know, I will argue that the naive application of this approach may lead to incomplete or even inaccurate interpretation of the data. For example, you can consider the case of a population of conjunctive neurons. Each one of them is tuned to some preferred combination of values for two variables. Now, if I know about or look for the coding for only one variable, I can calculate very nice tuning curves while completely overlooking the coding for the other variable. This scenario is not at all hypothetical. For example, in my PhD work together with Professor Nachum Molonovsky, I recorded neuronal activity from the hippocampus of crawling bats that crawled on a 2D floor, and I saw that conjunctively to the tuning the cells to position, something that was very well known, they also encoded at the same time the head direction of the animal, a fact that was overlooked even though it was studied for many years. A second example is the averaging over perceptual or cognitive errors. Consider, for example, a rat that's running back and forth on a linear track, and we look at the neuron activity from its hippocampus. And as Yaniv showed you yesterday, these kind of cells are tuned to position. So let's say we record from two neurons, and we see that each one of them has some preferred position along this linear track. However, let's say we record from the same rat, the same neuron, the same familiar environment on another day, but on that day, the animal is tired or less attentive, and let's say 30% of the time, it loses its orientation. And sometimes when it's at the left end and running rightward, it thinks that it's at the right end and running leftward. And then if we just naively calculate what is the average firing rate of each neuron at each given position, we'll have this kind of result. It will look, and we may get the wrong interpretation that there was something different now about the coding scheme of the neurons, that it used to be single peaked and now it's a double peaked, but this is not the case. Nothing changed about the coding scheme of the neurons. The thing that was changing now is just the perception of the animal, or the anchoring of this perception to the external state. A third example might be multiple neuronal representations for a given external state. Let's say that we have three external states that we can measure as an outside observer. A blue state, a red state, and a green state. However, each one of those states has two different neuronal representations. Here I consider the neural representations when I look at two neurons simultaneously. So if I just naively calculate what is the average firing rate for each one of the neurons given each external position, I may have a very wrong conclusion. I will have an average representation that is not representational at all. It's not. It doesn't look like any pattern that appear in the brain when this stimulus is observed. And I can also get here the wrong conclusion that the blue stimulus and the red stimulus have very similar representations, and that is different from the green stimulus. This is also not an hypothetical case. Specifically, in recently published work from our lab, we saw that when mice repeatedly visiting the same familiar environment, this same environment can have some repertoire of representations, and on different visits, different members of this repertoire can be used. So what we wanted to ask is, can we expose the key features of internal representations by analyzing brain activity alone without relying on behavioral level measurements? And this way, to circumvent some of the limitations of the classical approach that requires a priori assumptions regarding the identity of the encoded variables. So our approach will be to first determine the structure of relationship between the neuronal population activity patterns, and then maybe try to understand something about the computational process or the coding scheme from this structure for itself. And then to characterize the activity of single neurons with respect to the structure rather than relative to any preselected external variable. Okay, so I guess Yaniv showed you this kind of thing yesterday, but toward addressing this question, Nitzan Geva from our lab trained mice to run back and forth along a linear track to collect water rewards at the ends. And throughout the session, we used calcium imaging to track neuron activity in the hippocampal CA1 neurons. Here you can see the neuronal activity in the field of view as can be measured using a miniature microscope. And it was recorded as the mouse was running. You can see here in the lower panel. And here I marked 6 different cells. And in the middle panel, you can see the position of the mouse when each one of these cells were active. So you can see that each one of these 6 cells has different preferred position. It tend to be active when the mouse was at a given position along the linear track. So this is what people call place cells, and it's not surprising at all to find them in the hippocampal region CA1. Okay, so we can just count what was the time spent at each position along the track, what was the overall activity from a given cell at each position along the track, and then divide them and get the average activity rate for each position, namely the tuning curve of the recorded cell for position. I just want to say that although this kind of analysis yield very nice maps that show the tuning of all cells to position along the track. Here we just sort the cells according to their preferred position. It is important to notice that these maps were obtained via a very specifically tailored analysis that was developed within our paradigm over decades. For example, we limit our analysis only to a subset of neurons, namely place cells. We use a somewhat arbitrary threshold, such as including only running epochs in our analysis. We separately accounting for neuronal activity at different running directions. And maybe most importantly, we restrict our analysis to a single predefined variable, which is in this case position or location along the track. Okay, so what can we do differently? Here we analyze the data from 15 minutes long sessions in which mice ran along the linear track while we image the neuron activity in the C1, just like I showed you in the movie. And then we constructed neuronal activity vectors. Each of them constitute the activity of all cells in the data within a single 50 milliseconds time frames, regardless of what the animal was doing at that time. So each vector is of length n, where n is the number of neurons, and each entry is either 0 or 1 based on whether this neuron was active or not at that time frame. Okay, so in the data I'm going to show you, we had more than 450 cells. It is not very easy to work with such a high dimensional data. So we applied dimensionality reduction method, specifically Laplacian eigenmaps, which is a non-linear dimensionality reduction technique. And we apply it to all the vectors from this recording session, and this is what we got. Okay, so here each data point represents a single time frame in the leading 3 dimensions of the reduced space of neuron activity, which obtained from all neurons that were active during this session. And it is important to me to mention in this context that I'm not claiming that the results that I'm going to show you here are unique to specifically to the dimensionality reduction that I use, Laplacian eigenmaps. However, I would argue that that here it is important to use a non-linear a method rather than a linear one, such as principal component analysis (PCA), that is very commonly used in neuroscience. Okay, so if we look on the structure of the data in the lower dimensional space, we find high density of data points within a small number of clusters that we labeled A, B, C, D, and E. Okay, now the network seems to leap between these clusters or states in a periodic pattern, and I want you to notice that one of these states, and only one, appears twice within a cycle. And when we look at the neuronal data that we from the different sets, we found that the epochs in which the network was in state C actually consisted of two different states that appear here in red and blue. And as you can see, they also match the two phases of appearance of these states within a cycle. So we turn these two substates C1 and C2, and based on the transitions between all states, we can portray the typical dynamics within the structure, going from A to B to C1 to E to D to C2, and then to A again. Okay, for another cycle. Okay, so in everything I've shown you so far, we considered only the neuronal activity data and not the behavior of the animal. But now we can go back to the behavioral data and look what the mouse was doing during each of these neuronal states. Okay, so when we concatenate all frames of the behavioral movie from epochs that the network was in a given state, for example, here in state C1, this is what we see. The mouse was running leftward, okay, in this box, and I want to emphasize that this is not the same clip in repeat, but different epochs of the network visiting state C1, okay? And now what about, say, state C2? In that state, the mouse was running rightward. In state A, the mouse was drinking at the left side of the track, and on times that the network was in state D, the mouse was turning at the right side of the track. It is also nice to see that the symmetry of the track or behavior is reflected in the symmetry of the internal structure of neuronal activity. This way, A and E are the states of drinking at either left or right side of the track, and B and D are the states of turning at the left or right side of the track. Okay, so having found that different states of neuronal activity correspond to different behavioral states, we wanted to know if we can use the structure of the data to identify behavior in a finer resolution. Okay, so toward this goal, we focused on states C1 and C2, the states that were associated with running, and we went back to the data at the level of the neurons, namely before the dimension reduction, and then we dimension reduce only the data from the time that the network was at a given one of those two states, okay, separately to each one of them, and this is what we got. We got that the patterns from these states created this continuous structure or a manifold. Specifically here I colored the data points according to the position of the mouse, but this is just for visualization. I did not use that for the analysis, but you can see that the position of the internal state of the global network along this manifold actually corresponded to the physical position of the mouse along the linear track. We use the subclustering in order to parameterize this structure, and this allows us to reconstruct the trajectory of the mouse along the linear track. Here, the states of the network activity, namely the different subclusters, shown in magenta, and you can see that they corresponded quite nicely to the trajectory along the track that shown here in black. And we also quantified here in the subset in the subplot the disagreement between the two up to the single degree of freedom, which is the symmetry to a left-right reflection. And as you can see, the mismatch is quite narrowly distributed around 0, very different from what we get from shuffle data shown here in green. Okay, so given that we know the internal state of the global network at each moment, we can now go back to the level of individual neuron and define the tuning curve of each neuron, not with respect to any predefined external variable, such as place, but with respect to the internal state of the network, namely what was the activity level of a given neuron for given states along, for example, this curve for manifold. Okay, so when we do that, we get internal tuning curves that was very similar to those that were obtained with respect to position along the linear track. The tuning to position along the C1 structure corresponded to the position of the mouse during leftward running, and the tuning to the specific state along the C2 manifold corresponded to the mouse position along the rightward running. And I want to emphasize again that these internal tuning curves were obtained solely from the structure of neuronal activity and without relying on the behavioral measurements. Okay, so to conclude so far, we saw that the internal structure of neuronal population activity allows extracting neuronal tuning curves from brain activity alone. However, we still want to know to what extent does the internal structure of neuronal population activity merely reflect the behavioral task? For example, here we have a task and behavior that are symmetric to reflection, and so was the internal structure of neural activity from the hippocampus. Would that be the case for any brain region? And secondly, can similar analysis reveal internal representations using recordings from brain circuits that have not been associated with a specific canonical encoded variable? And when I say canonical, I mean such as space for the hippocampus, or orientation of the visual stimulus in primary visual cortex, the pitch of the tone in the auditory cortex, and so on and so forth. Okay, so to this end, we recorded from the anterior cingulate cortex. This is a frontal area that is associated with many higher functions, and not with a specific canonical variable. We conducted calcium imaging in the ACC of freely behaving mice that performed the same linear track exploration that I described for the hippocampal imaging, we applied a similar analysis to what we did there, and this revealed a quite different structure than what we saw for the hippocampal data. For example, you can see here that this structure is lacking the symmetry to reflection that the structure of neural activity in the hippocampus had for the same task. So we clustered the data points into different network states. This can be seen here in the different colors, and we saw a recurring cyclic pattern of transitions between these states. However, you can see that here within each cycle of or each behavioral cycle of running back and forth on the linear track, the network transitioned twice through all the states. Okay, like here. Yep. And this is very different than what we see in the CA1, in the hippocampus, where one behavioral cycle corresponded also to a single transition through the different network states. Okay, and now, like we did in the hippocampus, we went back to the behavioral data and sorted it according to the network states, and this is what we got. Okay, so we see that for each state, each state corresponded to a distinct behavior. Like the first state corresponded to turning, but regardless of the position along the track, the second state corresponded to the beginning of the running epoch. Again, unlike the hippocampus, regardless of the direction, the third cluster is the end of the running epoch. The fourth cluster is the drinking. Again, this is not the same flip and repeat, just different epochs of the network visiting this state. And the last state or cluster corresponded to rearing states where the mouse was standing on its back legs and looking around. Okay, so then we confirmed this clustering-based classification using manual labeling, and you can see the nice correspondence between the two, although the upper one was obtained using unsupervised analysis of the neuronal data, and the bottom one was obtained using manual labeling of the behavioral data. Okay, and as you can by now maybe expect, we find that in the ACC, but not in the CA1, the neuronal activity during epochs of the same behavioral states was similar at opposite sides or directions of the linear track. Okay, so if we now focus on the networks that were linked to running, we find that the neuronal activity represents the position of the mouse relative to the start and end points of each track traversal, regardless of the running direction. Okay, and we call that a trajectory phase. Okay, so here the data points are colored according to the trajectory phase, and that is the distance of the mouse from the start of the track, and we consider opposite ends of the track for the opposite running directions. And just like we did for the hippocampus, we constructed internal tuning curves and found that the internal tuning captured the external tuning of the same neurons to the trajectory phase. Okay, and just to further illustrate that, even in the level of a single neuron, this is a trajectory phase cell from the ACC. In blue, you can see the trajectory of the mouse as it was running back and forth along the linear track as a function of time. in green and red, you can see the position of the mouse at the time that this specific cell was active during rightward and leftward trajectories, respectively. So you can see that this cell had some preference to the 2/3 of the track, regardless of the running direction, and this is very different than what we typically see in the C1, in the hippocampus, for a familiar environment. Typically, what we see is a preference for a given position in a given direction, and if there's also tuning to the other direction, it will be at some other arbitrary position. Okay, and this is true, of course, not only for these two example cells, and also at the population level. Here I show you the population level correlation across the patterns for all different positions at the two running directions in the ACC. You can see here these secondary diagonals that indicate the similar that indicate the similar coding for opposite positions along the track for the opposite running directions, and this is very different than what we see for the hippocampus, where these secondary diagonals are completely absent. Okay, here are just three example mice recorded from the ACC. Here I colored only the data points during running according to the trajectory phase, and you can see the nice continuum of trajectory phase in this rainbow-like pattern. Okay, so just to conclude this point, we saw that in different brain regions we can see different internal structures of neuronal activity, even under the same behavior or task, and this possibly may reflect the different coding schemes of those different brain regions, or even the different computational processes they realize. Okay, just to further demonstrate our ability to study neuronal code regardless of or without using behavioral measurements, we tend to use publicly available data from the lab of Yuri Bujaki, where Adrian Telash recorded using electrophysiology from the postsubiculum and antrodorsal nucleus of the thalamus, brain regions. Those are two brain regions that are known to be associated with the encoding of allocentric head direction. And when I say allocentric, I mean that this is the head direction not relative to the body of the animal, but relative to the environment. It is not magnetic, but you can think about it as north, west, east, and south, etc. Okay, so they recorded the neural activity from neurons in these brain regions when mice really explored the two-dimensional open arena. Okay, and then we together with Orpin Khasov, a master student from our lab, we analyzed this data in the same way we did for the two other data sets, and here we got internal structure that has shape or topology of a ring. Okay. Here I colored the different data points according to the actual head direction of the mouse, and you can see the continuous trajectory of the internal state of the network along this structure in the reduced space in the reduced space of neural activity or manifold of or neuronal manifold, if you will. Okay, the head direction also can be indicated by the clock in the lower panel. Okay, so the continuity of the trajectory of neuron activity can be also seen by the fact that when we cluster the data points, the transitions across these clusters were only between neighboring ones. So we can then use the clustering to parameterize the internal state of the network, okay, and so that up to the internal symmetries, which are in this case the symmetries to rotation and the reflection, the internal direction or angle, namely the position of the state along this manifold, shown here in red, is in agreement with the actual head direction of the animal, and the mismatch between them is again shown in this sub panel, and you can see that the errors are quite nicely distributed around the zero. Very different from the shuffle data in green that was also corrected to optimize the degree of freedom for reflection and rotation. And then now we can go back to the level of a single cell and ask what was the activity of each given cell as a function of the internal state of the network along this ring, and we saw that the tuning curves of the cells to this internal variable very nicely corresponded to their external tuning to the external variable, which is in this case the head direction. Okay, I just want to mention that a very similar approach in using the very same publicly available data was taken also in the lab of Ila Fit and in the lab of Benjamin Dunn, and they also published recently papers making very similar conclusions. Okay, so since this approach does not rely on behavioral measurements, we turn to test it in case that the behavioral measurements is irrelevant, okay? Namely during sleep, when the head direction is mostly constant and in general is not corresponding to the neuronal activity in these brain regions. So when we apply the same analysis on data that was recorded in the same mouse during REM sleep, this is what we got. So we can see here that also for the data from REM sleep periods, when we reduced it, we found that the data had a structure with a ring-like topology. Okay. This result did not come as a total surprise to us, since it is consistent with Peyrash's finding that pairwise correlation during wake periods are maintained only during sleep. Okay. So also here we can go back to the single neurons and see that the internal tuning of the neurons that was obtained relatively to the internal state during sleep corresponded to the tuning of the same neurons to head direction during awake period. Okay. A common practice is to devise a decoder based on awake data and then to apply it on data recorded on sleep periods, and then obtain some virtual trajectory of the animal or something like that. However, the performance of this kind of decoder cannot be validated since there is no measurement of ground truth. So here we took a different approach, and instead we used the activity patterns during REM sleep, and based on that, devised a decoder to infer the head direction of the animal during awake periods, which can then be validated against the actual head direction. Another way to put it is to say that differently from what usually decoders does, where you have like a training set that contain both the data and the labels, here we had labels for the test data, but not for the training set. Okay, and yet we obtain very nice results. Okay, yeah, I want to acknowledge that properties of the internal structure here can be just seen when visualizing the data. Yeah, you can say, okay, so it looks like a ring, it looks like one-dimensional manifold, but these properties can be also estimated analytically. And I would like to dedicate the next few minutes to discuss these methodological issues. Okay, so let's consider a case where we have data, and let's say that this data is one-dimensional. And then one thing I can do is ask, given the data point, how many neighbors it has, namely how many other data points are there within a sphere around this data point. Okay, so the answer to that is obviously depends on the size of the sphere. However, as I increase the size of the sphere, the number of the neighboring data points would increase. And in the case of one-dimensional data, this number would increase linearly with the radius of the sphere. In the case of 2D data, for example, this would increase as the square of the radius. And in general, the number of neighboring points with radius r would grow as r in the power of d, where d is the dimension of the data set. Okay, at least when we consider relatively small range of radii. Okay, so here y is the overall number of neighbors, c is some density, r is the radius, and d is the dimension of the data. Okay, so we can also take the log of the two sides of this equation, and this tells us that if we plot the number of neighbors and the radius in a log-log scale, the slope would just indicate this intrinsic dimension of the data. Okay, I think this is also called a correlation dimension, and I think that you saw a somewhat similar definition of dimension, the box counting, that also called the Minkowski dimension, and there are also a definition like the Hausdorff or information Hausdorff's dimension. There's also the information dimension, and they are somewhat similar. There are cases where these definitions are not overlap, but in many cases there is agreement between the different definitions. Okay, but the point that I want to emphasize here is that these definitions of dimensions are very different than the notion of embedding dimension. Okay, that is very commonly used in many fields, and specifically in neuroscience. Usually, we're using using PCA, and then asking how many components we need in order to capture 100% of the variance of the data. Okay, so here I show you two examples of simulated data. Both are very high dimensional, but you can see that that the embedding dimension of each one of them is 3. You need only 3. In either case, you need only 3 dimensions, 3 components, sorry, in order to capture all the variance that you see in the high dimensional data. Okay, namely, it can be spanned using using a basis only with 3 vectors. However, if I look at what is the intrinsic dimension of these data sets, I can see that while data 1 has an intrinsic dimension of 1, data 2 has the intrinsic dimension of 2. And this is because the first data set is kind of a banded ring, and the second data set is a sphere, yeah, like the outside of a 3-dimensional ball. Okay, so so they both need like 3-dimensional space in order to embed them, but the internal dimension of the ring is the intrinsic dimension of the ring is 1, and of the sphere is 2. Okay, you can consider the embedding dimension as the dimension of the space that one needs in order to contain the object, and the intrinsic dimension as the dimension of the material that the object is made of. Okay, you can also when calculating it, you can have some correction for non-uniform density or for curvature. You can also maybe it's important to say here that you can also obtain that in some cases if you do a local PCA. And just as an illustration, here is a picture I took from some street art in Brooklyn. So you can see that this object has the embedding dimension of 2 or 3, but the intrinsic dimension is 1 because it is made of a metal wire. Okay, so when we look at our data, we can see that in the case of the head direction data, the embedding dimension is 1. You can see here the slope 1 in the log scale, and that was also the case for either the internal structure in C1 and C2, namely the structures for leftward and running epochs, as you would expect for running for coding of position along a 1-dimensional corridor. Okay, so I just very clearly there is more than just the intrinsic dimension. For example, you can consider a segment and the ring, like we have in those 2 cases, that both are 1-dimensional, but they are different because they have different topology. Okay, and therefore, so you saw some examples for that, but yet I would very shortly repeat some of the concepts. We can apply topological data analysis in order to study the topology of our neuronal data, or of its internal structure. Maybe the simplest topological property is the number of separate components. Okay, so let's say just a simple example: you have a data set that consists of 40 data points, and you take a look and you say, okay, it seems like I have here two clusters, two components. And then I can come very rigorously, or but very naively, and say, what do you mean by two components? I see here 40 points. So from topological point of view, you have 40 components here. There's nothing here that supports two components. Then you can say, yeah, I see your point, but try to build a sphere around each one of the points, and then consider any pair of points that have overlapping spheres and as connected, and then you can consider the graph that you can, and then you see that you have two separate components within this graph. And then I can quite naively say, yeah, this is a great idea. I'm going to do that, the same thing. And here I build a sphere around each one of the data points. I consider which pairs of points are connected, and say, yeah, I have in this data set one component that are all connected. And then somebody can tell me, yeah, but it seems that you chose the radius of the sphere to be too large, and say, yeah, but what do you mean? I chose the radius to be too large. How should I choose it? And then the answer, as you already saw, is not to make a specific choice for the radius, but instead to just consider all radii, namely to gradually increase the radius and consider the graph, and therefore the components for each radius for each radius in the relevant range, and to see which in this case separate components are stable or persistent through a relatively wide range of radii. Okay, this is also called this method is called barcode. Okay, so just to make it very explicit, here we can see that there is a very wide range of two persistent or stable components. So all that will be always the case, that if we go for large enough radius, we'll have one component, and usually when we go for epsilonic radius, we'll have more or less the number of components as the number of our data sets, of our data points. So, now we can, and for every given radius, we can define we can associate the data set with a specific graph, and then we can also associate every click of k plus nodes, namely k plus nodes, that data points that are all connected to each other, as a kth order simplex. And this way we can consider more things like higher when we consider the higher degree simplexes, we can find higher degree features such as caveats of different dimensions. Here, for example, you can see a hole for this specific value of the radius, but this hole is quite unstable. If you go for a bit smaller radius, you will not have this edge, for example, so you will not have the hole. And if you go for a bit higher radius, this hole will be covered with triangles. Yeah, with third order cliques. But when we apply this same approach for our analysis of the data from the head direction, in the upper panel it's a simulation, but the lower panel is the actual data. We can see that it has one component, one connected component, one hole. Sorry, one hole and zero spaces. Okay, you already heard it, but these are like this can be associated with the Betty numbers of the topology of the structure. So and here the Betty numbers 1, 1, 0 is consistent with a ring, or at least ring is the simplest topology that can be associated with these values of Betty numbers. And as I said, for this case, I can just eyeball the data and say that it looks like like a ring. But it's important to be able to also analyze this thing analytically and using these tools to estimate the structure for the cases when the data could get richer and the topology would get more complex, and it will be just harder to infer the dimension of the topology from simple visualization. Maybe a first step toward this direction is in a very nice preprint from the lab of Maybrit and Edward Moser, where they showed that the neuronal activity from a single module of the neuronal cortex has the topology of two-dimensional torus. Okay, so now let's go back to the calcium imaging and to the hippocampus, and Yaniv already showed that, but we know from his work together with Mark Schnitzer, where he did his postdoc in Stanford, and we know that when the mouse explores the same familial environment, namely repeatedly visiting the same environment it already know, upon different visits, somewhat different subsets of the neurons are active and encoding space. Okay, so naturally we wanted to find out to what extent would the internal structure of neuronal activity be stable over time, given this substantial turnover in the active ensemble of cells across days. Okay, and the thing is that if the internal structure of neural activity reflects the computational processes undertaken by the network, then this structure should be invariant property. Okay, and indeed, what we found is that despite this major turnover in the identity of the cells that are active upon different days, the structure of neuronal activity is similar. Okay, so and what is nice is that the internal structure is conserved not only between different days of recording in the same mouse, but also across different mice that were trained to perform the same behavioral task. Okay, so that allowed us to devise a decoder that infers the position of one mouse based on the data from another mouse, again up to the symmetry of reflection between left and right. Okay, so and I just want to make it crystal clear that here in this decoder, unlike the common decoding methods, we will use one set of like one data sets with a given neurons to devise a decoder that will be tested on a different, separate neuronal population. Okay. So the actual procedure we use here is very simple. Okay. We look at the pattern of activity for a mouse, and let's say this is a mouse that we have its neuron activity, but we never saw its behavior. But because we have its neuron activity, we can find the internal structure of that activity, and we can tell with what part of this structure, or what is the state that is associated with this activity pattern. Okay, and we saw that this internal structure of neural activity for that task is similar to the structure that we have for another mouse. So let's say that this is a mouse that we know also its behavior, so we can find an analogous state within the structure of that mouse, and then we know and we can tell what was the external state of the mouse or behavior that was associated with this internal state, and that would be our guess or estimation for the meaning of this pattern of neural activity in mouse 1. And you can see that we had a very nice performance for this decoder, both across days for the same mouse and for data across individuals. So I'll just like to show you a short clip that will further illustrate this procedure. Okay, so here in the upper panel, this is the internal structure of neuron activity from a mouse 1. Okay, this is its behavioral data, and I'll mark here in red point the pattern of activity in the hippocampus of this mouse at any point in time. Okay, so here in the upper right panel, this is the internal structure of neural activity from another mouse, and you can see that those structures are very similar, again up to the symmetric reflection. And based on this similarity, I can, at any point in time, choose a data point from an equivalent state or part within this internal structure. And because I know also the behavioral variable activity or variable from this mouse 2, so I know what it was doing at the time of that activity pattern in the hippocampus. So I'm going just to put here the behavioral frame at that time. Okay, so just show you the clip now. Okay, so you can see it's a bit jumpy and it's not perfect. But yet, at some level, we can reconstruct the the behavior of one mouse, or of one mouse, only based on the on what we know about the relationship between neuronal activity and the behavior in another mouse, because we use the similarity between the internal structures of this mouse, and you also use the assumption that the equivalent parts of these structures has equivalent semantics. They correspond to similar external state. Of course, as I said, this is not perfect, but I hope that with better recording methods and meta analysis, we can have both better signal to noise ratio in the decoder, but also maybe be able to decode richer and richer behaviors. Okay, so just to summarize, I show you that the internal structure of neuronal population activity allows extracting neuronal tuning curves without a priori assumptions about the identity of the encoded variables. I show you that for behavioral state and position along the track in the hippocampus, for behavior and trajectory phase in the ACC, and for head direction in the postsubiculum and the ADn. Also, we saw that the internal structures differ across brain regions, even under the same behavior. This may provide a fingerprint for their underlying computational processes or coding schemes. Specifically, we saw it for the C1 subregion of the hippocampus and the ACC, where this reflected the fact that one encoded the position and the other one the trajectory phase. We suggest that as an approach to study the conservation of the structure of network activity in a way that is indifferent to the specific identity of the neurons. And indeed, we see that conservation of a structure across mice and over time reflects stable stereotypic relationships between patterns of neuronal ensemble activity within the recorded population, even when the recorded neurons themselves are different. Okay. So at this point, I would like to thank the organizing committee. I'm really honored to take part in this school. It is only a bit misfortunate that this is the Corona times, and I have to miss the side talks and with the students and the other speakers, which is always the most interesting part of schools or conferences. And I would also like to thank Yaniv Ziv and all the people in the lab, and specifically to Nitzan Geva, Liron Sheintuch, Yoav Rechavi, Noah Eilat, and Or Pinchasov, who all took very important and active parts in this project. And of course, you for listening. Thank you very much.